Today we are going to discuss Edexcel Unit 5 paper. So I'm going to start with the question number 7. And question number 7 is about the genetic engineering, and it says animals and plants can be genetically modified, or recombinant human erythropoietin is a protein drug that can be given to patients with a low hemoglobin concentration. Sheep have been genetically modified to produce recombinant human erythropoietin in their milk. Explain how a sheep could be genetically modified to produce RHE in its milk. Okay, to understand the concept and to the answer for this question, the genetic modification we can do for bacteria, fungus. Here's the bacteria. We can genetically modify bacteria, and this is a yeast cell, fungus, and we can also genetically modify plants, and we can also modify animals using a recombinant uh, using gene technology. So if we want to modify bacteria or yeast cell, plant or animal, we have a general protocol. We can follow the general protocol with slight modification. So first I will introduce the general protocol what we can use. This question asks about the sheep. Now if we want to modify the sheep genetics, so the first thing we can do, we have to isolate the human gene, RHE gene. Isolate the human RHE gene. Now how we do that? We can cut this gene using restriction endonucleases. That's the first process, cut RHE gene using restriction endonucleases. Okay, so if we start with the first process, that's a human cell, and we have this gene now, that's the RHE gene. Then the next stage, we want to introduce to the vector, because we cannot introduce the piece of DNA to the cells. Vector can deliver efficiently. For example, the common vectors we can take the plasmid. So plasmid is like this, and we can use again restriction endonucleases, and we can insert the human gene to the plasmid. Alternatively, we can use the virus, and we can introduce again RHE gene to virus, and these vectors deliver the gene to zygote. So after deliver the gene to zygote, now this zygote from the animals, and they can express this gene in the mammary glands and the milk containing RHE protein. So this is the basic protocol we use for genetic engineering, and cut the gene using restriction endonucleases from human cell. Then the second, insert the gene using a suitable vector such as plasmid or virus. After that, allow the plasmid or virus to deliver to the zygote. Then zygote express the gene to produce a recombinant human erythropoietin. This appears in the milk. So the four steps we can write to get 4 marks.